Hello everyone. Today we will learn about nanoparticles. Today is an interesting topic we will learn. So today's topic is nanoparticles. It is a very much important topic in novel drug delivery system. Okay. So what are nanoparticles? They are actually used as a novel drug delivery system or kind of specific drug delivery system to deliver the drugs to a specific tissues and cure various kind of disease. So what they do? They deliver drugs to specific tissues and cure various kind of disease. Okay. Now, why they are named as nanoparticles? Because the materials which are used in such system available with a dimension under 10 to 1000 nanometer. They have a dimension about 10 to 1000 nanometer. That's why they are known as nanoparticles. Why we use nanoparticles as a drug delivery system? Because it allows faster drug absorption. In a controlled manner and minimized side effect. Now, what are the ideal properties? So, what are the ideal properties of nanoparticles? First one is it should be stable in physiological pH. Or in blood. The second one is must be biodegradable. And the third one is non-toxic to living tissues. Okay. Now we will understand about what are the types. So the next is types. Nanoparticles are available in two forms. First one is nanocapsules. First we will understand about nanocapsules. Nanocapsules are such system in which drug is present in the inner central liquid core cavity which is surrounded by a solid polymeric cell. Okay. So, drug is present in the liquid core cavity which is surrounded by solid polymeric cell. Now we will understand it through a diagram. So these are the drugs present in the liquid pore cavity. Okay, this is the liquid pore cavity and surrounded by polymeric cell which is solid. Okay. Now the second one is nanospheres. What is nanospheres? In this system or in such system, drug is dispersed throughout the polymeric matrix or adsorbed on the outer surface. So the drug is dispersed throughout the polymeric matrix or it can be adsorbed on outer surface. Now we will understand it through a diagram. So this is a polymeric membrane. And these are the drugs. These drugs will remain in the dispersed form or it can be adsorbed on the outer surface like this. Okay. Now, which type of polymers we used for such system? Coming to the next, which type of polymers? So, polymers used. Polymers are based on two types. First one is natural polymer, which is hydrophilic nature. Shows hydrophilic nature. Okay. In the natural polymer, albumin, gelatin kind of proteins are used. These are proteins and alginate, Chitosan, Agarose type of polysaccharides are used. Okay. Now, the second one is synthetic polymer. Which is hydrophobic in nature. For this, polystyrene, type of polymerized polymer used, or sometimes Polyisobutylcyanoacrylate type of polymerized in-process polymers are used. Now, what are the various methods? So, the methods of preparation. Methods of preparation. First method is preparation of nanoparticles by polymerization technique. In this technique, two type of polymerization is used. Process is used. First one is emulsion polymerization, and the second one is interfacial polymerization. Okay. Now, the second method of preparation is preparation of nanoparticles using only polymer, okay, not the polymerization technique. So in this category, the methods are, first one is solvent evaporation, method, and second one is spontaneous emulsification, or solvent diffusion method. Now the third method is Ionic gelation or Coacervation of hydrophilic polymer. And the last one is supercritical fluid technology. Now, what are the advantages and disadvantages of such type of system? First one, advantages. Advantages are drug carrier have higher stabilities. So, higher stabilities. Second one is higher the carrier capacity. So, higher carrier capacity. Third one is feasibility of various routes of administration of both hydrophilic and hydrophobic substances. So, various routes of administration with both hydrophilic and hydrophobic substances. And the fourth one is it can store for longer period of time and reduces the dosing frequency. So can be stored for longer period reduces the dosing frequency. Okay. Now what are the disadvantages? So disadvantages are, first one is toxic metabolites may form because it can be stored for longer period of time. So obviously toxic metabolite can be formed. Second one is discontinuation of such type of therapy is not possible. So discontinuation is not possible. And the third one is limited target abilities, or it can target only specific sites. So limited target abilities. And the fourth one is obviously cytotoxic. Okay, so these are the advantages, disadvantages. So this is the end of lecture. If you like my video, please like, comment and subscribe. Thank you for your attention.